So this next section talks about the different types of mutations that can happen in bacterial cells. So first we're going to talk about what the difference is between wild type and the mutant strain. So wild type is the most common genotype that's found in nature. So typically when we're talking about wild type, this is the strain that does not have the mutation. The mutant strain has a mutation. There are different causes of mutations. Most mutations are spontaneous. So a spontaneous mutation happens randomly. And it's usually by an error in DNA replication. Induced mutations are induced by particular mutagenic chemicals. So you add a chemical to something and then you see a mutation happen. So there's certain substances that we can impose on bacterial cells that can cause them to mutate. And there's different categories of mutations. So a point mutation, let's say our DNA sequence was supposed to be AAATTT. A point mutation would be a change in one base, such that now we might have AAATTC instead of TTT. They involve an addition, deletion, or a substitution of a single base. A lethal mutation can lead to cell dysfunction or death, and a neutral mutation has no noticeable change, so it doesn't help or hinder the function of the bacterial cell. A missense mutation is any change in the code that leads to the placement of a different amino acid. If you remember when we talked about the genetic code, and actually let's go back and take a look at that genetic code because it's right up here. So here you see that UCU codes for serine, UCC also codes for serine. So if we had a point mutation that changed the last U to a C, it would still code for serine. But in a missense mutation, it's any change in the code that leads to placement of different amino acids. So going back to our code up here, if we had, for example, AAU and we changed it to AAA, well, now changing one base has led to the placement of a different amino acid. So AAU would have been asparagine, but since we changed that last base, now it codes for lysine. So it's a missense mutation when a new amino acid is put in. A nonsense mutation changes a normal codon into a stop codon that does not code for an amino acid. So if we had UAU and we were supposed to put in a tyrosine, and we had a mutation happen in this last U so that it got changed to a UAA. Well, changing to UAA would change that to a stop codon, and that would cut the protein chain short. So that's a nonsense mutation. A silent mutation alters a base but has no effect on an amino acid. So going back to code here, that's like this example, UCU, you can change this last U to C, A, or G, and you'd still code for the amino acid serine. And a back mutation occurs when a gene that has undergone a mutation reverses to its original state. So if it goes back to being infectious, for example. This is one of the risks that we have while working with live attenuated bacteria in our lab class. So we know that our bacteria that we use in our academic lab is non-pathogenic. but there's always a chance that it could mutate back to becoming pathogenic. And that's why it's a little bit dangerous to deal with some of our microorganisms. This is also why we practice universal precautions. So we treat every sample as if it is infectious and potentially deadly, so that we never pose a risk of getting infected with something that's really bad. So here we look at this illustration. So if the wild type strain is the one big fly had one red eye. The reason why they're 3-letter words is, again, remember, if this is a sequence of messenger RNA, it's read in series of 3 bases. The codons are 3 bases on messenger RNA. A substitution would just substitute like 1 letter. So the rest of the sentence would make sense. It's just this one substitution happened. Frameshift. A frameshift mutation can be one of the most dangerous types of mutations. If you add or delete bases that are not series of 3 bases, you have shifted the entire reading frame for the rest of the protein. So here, the 1 still makes sense, but you add this Q here, and now you have a different reading frame. So the rest of the protein doesn't make sense. So frameshift mutations are one of the most dangerous types of mutations that you can get. If you delete 3 nucleotides, so you delete a series of 3, here they're deleting the word fly. So you still have the rest of it makes sense, you're just missing one big word or one amino acid in the ultimate protein that this produces. If you insert 3 nucleotides, like you add in this word, the rest of it is still identical to the wild type, but now you have this extra word in here, so this symbolizes the addition of an extra amino acid. So some mutations can be worse than others. Frameshift mutations affects every single base after or every single amino acid after that mutation. So adding in something or deleting something that's not in a multiple of 3 can lead to a frameshift mutation.